Hi everyone, it's Mrs. Bustamante. Today we're going to talk about 1.4 and the properties of biological macromolecules. Basically, we're going to talk about the properties of their monomers, the things that make them up, and how those bonds connect. Let's go ahead and start with carbohydrates. We remember that carbohydrates monomer is sugar. And it's composed of carbon, hydrogen, and oxygen. And the type of sugar and how it's linked together is determined by the structure. And that structure determines its function. You're going to hear that a lot. How things are put together really determine how they work, essentially. What are some examples of carbohydrates? Well, sugars individually, starches, and cellulose, all great examples of a carbohydrate. Sugars tend to end in ose. If you have a monosaccharide, that means you have a simple sugar. It's just made of one sugar. That's all it is. Glucose, for example, is a monosaccharide. Disaccharides are going to be made of two monomers, two things put together. Di means two, like sucrose, for example. And then we can have polysaccharides that are really large polymers of lots of sugars put together. Starch is a really good example of this. You do not need to memorize the structures of the sugar, but it's good to know that they're classified by the number of carbons there are. So hexose, for example, has 6 carbons, is really glucose. Pentose would have 5 carbons, is ribose. And that's kind of good to note just how you might name them or talk about them in the future sometime. Proteins, remember the monomer of proteins is an amino acid, and they have CH, carbon, hydrogen, oxygen, and nitrogen. So we add that nitrogen in there. That's kind of a key component of proteins. They are very diverse molecules. They include enzymes. They can be involved in the structure of something. They are involved in carrying and transporting things, cell communication, signaling, receptors, defense, movement, storage. Really, proteins are pretty essential to a working cell in general. The polymer makes up something called a polypeptide or a polypeptide chain. That's what we get when we put lots of amino acids together. And proteins can be one or more polypeptides together. The protein chain folds and bonds together in specific ways based on how it's composed. And we get a complex 3D shape. This is hemoglobin here, and this is Rubisco, two pretty common proteins. And Rubisco is actually made up of 16 different polypeptide chains. Hemoglobin is made up of 4 in your blood. So you can see that when you put them into a 3D structure, it looks pretty neat. Now, what do we need to know about protein structure and how we build it? Well, we're going to build our amino acid sequence, and we're going to call that our primary structure. There are 20 different amino acids. You can see all 20 of them down here. And we don't have to memorize all 20 of those amino acids, but we do need to know their general structure and be able to identify them. So what do we need to know about them? They all have an amine group. All amino acids have that in common, and they have a carboxyl group. This carbon double bonded to an oxygen and this OH, that's your carboxyl group. They're also going to be bonded to a hydrogen, so carbon gets its 4 bonds. And then they have something called an R group or a side chain. And this is the part that changes. It can be hydrophobic, doesn't like water, hydrophilic, does like water, and it can also be charged depending on this R group, then is going to help determine how that protein eventually folds together and has its shape. And again, we're going to hear again that shape is going to determine function, how it's put together. That primary structure will help determine how it folds and builds and makes its structure. So you don't need to know all the R groups, but you do need to be able to recognize the difference in them. So proteins then have what we call directionality. We have a carboxyl, COOH end, and an amino end, which is our NH2 end. And when we bond them together, when we take monomers and put them together, we form what we call a peptide bond when those link together. So here you can see our OH and our H of our amine group through dehydration synthesis. And what I like about this picture here is you'll see you have kind of a big long chain of N and H's, but this part down here is all a little bit different, and that's your R group. That's your variance in those. We say that one peptide chain can grow in the same direction. So you're always going to see, if you're going from left to right or really any direction, they grow from your N terminus, where your amino group is, to your C terminus, your carboxyl group. And the R groups are going to determine how the protein folds. That's going to give it its structure, which will determine its function, what it does. Next, we have nucleic acids. Remember, nucleic acid monomer is a nucleotide. It has carbon, hydrogen, oxygen, nitrogen, and now we add in phosphorus. And the reason we're mostly familiar with these is because we know about DNA and RNA, and it stores our genetic material in these two things, right? So nucleotides have three parts. They have a phosphate group, they have a pentose sugar, either deoxyribose or ribose in DNA and RNA, and then they have a nitrogen base. That nitrogen base we typically represent with a letter. We'll learn more about it later. A, T, C, G, or U. And that pentose sugar is a 5-carbon sugar. It's ribose in RNA, and DNA is deoxyribose, and then it has a phosphate group, and that's really how we bond them together. So nucleic acids, DNA and RNA both have different structures, right? If we look at RNA, it's single-stranded, and here are bases. RNA has cytosine, guanine, adenine, and uracil, and DNA is double-stranded. It forms our double helix. They go in anti-parallel directions, meaning they match up, but they are opposite of each other. And you have cytosine, guanine, adenine, and thymine. So really your difference is thymine is in DNA and uracil is in RNA. Next, let's talk about lipids. So what's the monomer of lipids? Well, lipids are kind of unique. They really don't have a monomer like the other ones we've talked about. And that's kind of important to know a little bit about them and to recognize, because they can have lots of different shapes. They contain carbon, hydrogen, and oxygen, and they are non-polar typically. They're made of long, what we say are hydrocarbon chains, where saturated lipids, basically we have carbon and hydrogen all throughout this chain. If you look at this part, all of it is carbon and hydrogen. Everything in there is carbon and hydrogen. We say something is saturated, a saturated hydrocarbon, when we have only single bonds between carbons, and then everything else is filled with hydrogen in there. So these bonds tend to be more rigid and structured. Unsaturated lipids can have one or more carbon that's double bonded to itself. And when you see that, it gets kind of bent, and it's more flexible. Unsaturated fatty acids at room temperature tend to be liquids because they're more flexible, and saturated fatty acids at room temperature tend to be solids because they are less rigid and can't move as much. Some examples: fats, steroids, and phospholipids that we'll talk about a lot when we talk about our phospholipid bilayer. Here's a triglycerol with 3 fatty acids linked to a glycerol molecule. That's how we get triglyceride there. So saturated versus unsaturated. Unsaturated fats or lipids are gonna be this bent, and they tend to be liquid at room temperature. And saturated fatty acids, because they're basically in a straight line, they can stack up on top of each other. They form typically nice solids at room temperature. And so those are things like butter, for example, versus vegetable oil. Sometimes you'll hear about unsaturated is better for you than saturated fats, because saturated fats can stack up on top of each other and sometimes interfere with cardiovascular disease. So phospholipids, there's a special lipid. It's called a phospholipid, and we're going to talk a lot about it when we get to our phospholipid bilayer. So what do we need to know about it now? Well, we say it's amphipathic, which means that it has a hydrophobic part and a hydrophilic part. It has a polar phosphate head, that means it likes water, and it has a non-polar fatty acid tail, which means it does not like water. So it can interact with water in non-polar environments. It can interact there, which gives it a really nice bilayer effect that we'll talk about a lot later on. The other type of lipids we need to know about are steroids. They tend to be 4 fused rings of carbon or more. Different steroids can be attached with different functional groups to their rings, and different structure, again, creates different function, so they will do different things. Examples of this, cholesterol or your sex hormones, these are all examples of steroids. So hopefully now you have a better idea about the properties of monomers and the types of bonds that they form.